Today we'll talk about the dominant epistasis. What is dominant epistasis? Basically, epistasis simply means when one gene suppresses the expression of the other gene. Obviously, these two genes are located different gene loci. Basically, this epistasis, it is of two types. Epistasis, it is of two types. We have got two types of epistasis. One is called as dominant epistasis, then we have got recessive epistasis. Now, what is dominant epistasis? In case of dominant epistasis, the dominant allele of one of the gene suppresses the expression of either of the alleles, dominant or recessive of the other gene. So here it is the dominant allele which suppresses the expression of the other gene. For example, we have this condition. This is the dominant allele of one gene and it suppresses the expression of the other gene. This A gene, it suppresses. A gene suppresses the effect. A gene suppresses the expression of the other gene, that is B gene. So we can say that this is a dominant epistasis. It is a dominant allele and it has suppressed the expression of it. Simply we can say it masks the expression of the B gene, or it can be like this. We have dominant allele A and it suppresses, it masks the expression of the other gene. So this is also a dominant epistasis. That means in case of dominant epistasis, dominant allele of one gene suppresses the expression of either of the alleles. That is dominant or recessive of other gene. So in case of dominant epistasis, this is dominant allele, it suppresses the expression of the dominant allele of other gene. This is also a dominant epistasis. Here, this A gene, it suppresses the expression. This is a dominant allele, it suppresses the expression of the recessive allele of other gene. This is also a dominant epistasis. These are both these cases. This case is a dominant epistasis. This is also a dominant epistasis. Now, in case of dominant epistasis, we have the dihybrid ratio, it gets modified. The normal dihybrid ratio, that is 9:3:3:1, it gets modified to 12:3:1 in case of dominant epistasis. And also this dominant epistasis, it reduces the number of phenotypes in the F2 generation. That means this in case of this normal, this is normal dihybrid ratio, and this is epistatic ratio, dominant epistatic ratio, dominant epistasis. And also we have got 3 phenotypes in the F2 generation, while as we have got 4 phenotypes in the normal dihybrid cross. So we can say that there are 4 phenotypes in the normal dihybrid cross, and here we have only 1, 2 and 3 phenotypes in the normal, in case of dominant epistasis. So dominant epistasis, this dominant epistasis, the ratio is 12:3:1, and also it reduces the number of phenotype types in F2 generation. I will explain this dominant epistasis. We'll explain it by means of an example. We'll take fruit color in case of cucurbita pippo explanation, that of dominant epistasis, and we'll take fruit color in cucurbita pippo. This, it belongs to family Cucurbitaceae, simply a pumpkin. It is called as summer squash. Commonly it is called as summer squash. This cucurbita pippo has 3 types of fruit color. That means it can be white fruit color, 3 types of, then yellow fruit color and green fruit color. So in case of cucurbita pippo, what is called as commonly summer squash, it has got 3 types of fruit color. One fruit color is white fruit color, yellow fruit color and green fruit color. This the fruit color in case of cucurbita pippo, what is called as cucurbita pippo, what is called as summer squash, it is governed by 2 genes. One is W and Y. These are governed by 2 genes. The fruit color in cucurbita pippo is governed by 2 genes. The fruit color in cucurbita pippo is governed by 2 genes. One is W and another is Y. So we'll make a cross which will help us to understand what this dominant epistasis is. A cross was made between, so we have got, if we'll take pure white, it is W and capital Y. And then we have got green, it is double recessive, that is small w, small y. We will make a cross between, we'll take 2 parents, pure white color. We'll take 2 parents, pure white color summer squash and we'll cross it with the pure green summer squash, pure green color summer squash. We'll take two parents. In order to understand what this dominant epistasis is, we'll take two parents. This is pure white summer squash. It will be crossed with pure green summer squash. So genotype is capital W, capital W, capital Y, capital Y. And this is homozygous double recessive. This green color summer squash is double recessive. So small w, small w, small y, small y. Then we have gametes. The gametes. These are simply gametes. Law of segregation. Then F1 generation. Capital W, small w, capital Y, small y. Obviously, this is F1. This is first filial generation, and this is white color summer squash. White color summer squash. And then we'll make selfing of F1. That is selfing. F1 individuals are crossed with each other. So we have W, capital W, small w, capital Y, small y. It is crossed with capital W, capital Y, small y. And we will get F2 generation. So we have gametes, male gamete, female gamete. So we have capital W, capital Y, capital W, small y, small w, capital Y, small w, small y. These are the gametes. Same is the case, we have capital W, capital W, capital Y, capital W, small y, and small w, capital Y, and small w, small y. So then we'll make a cross. There is the fusion or fertilization of these gametes. These are male gametes, female gametes. So we'll get W, 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 Y, Y. This is white color summer squash. W, W, capital Y, small y. This is also white color summer squash. This is W, small w, capital Y, capital Y. This is white, capital W, small w, capital Y, small y. This is white color summer squash. Then we have got capital W, capital W, capital Y, small y. This is white color summer squash. Then we have capital W, capital W, capital W, small y, small y. This is white color summer squash. Then we have another capital W, capital Y, small y. This is also white color summer squash. This is capital W, small w, small y. It is also white color summer squash. Then we have capital W, small w, capital Y, white color summer squash. Then we have capital W, capital W, small capital Y, small y. This is white color summer squash. Then we have small w, capital Y. This small w and small w, capital Y. This is yellow color summer squash. This is yellow color summer squash. We have small w, capital Y. This is also, we don't have capital W here, so it is yellow color summer squash. Then we have capital W, capital Y, small y. This is white color summer squash. This is capital W, small y. Also white color summer squash. Then we have small w and capital Y, small y. This is what yellow color summer squash. And then we have homozygous, double homozygous recessive, that is double recessive, small w, small y. This is green color summer squash. So we have here, we have, okay, write it here, we have white color summer squash is to yellow color summer squash is to green color summer squash. How many white are there? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. We have 12 white summer squash. How many yellow? 1, 2 and 3 yellow color summer squash, and green 1. So this is 12:3:1. Remember that this W gene, it is W gene, or we can say that this gene, it is epistatic. It is epistatic to Y gene. So we can say that it shows, this shows, or capital W, small w, this shows white color. That is W gene, dominant gene, that is dominant gene W. It prevents the expression of Y gene. It masks dominant gene W, it masks Y gene. That means, it means that in the absence of W gene, this Y gene expresses, and we have, therefore we have this small w and capital Y. It produces what color? Yellow color. Same is the case, we have small w and capital Y, small y. It produces yellow color. That means, whereas in case of the capital W, small w, capital Y, small y, it produces what? White color. Or we have capital W, small w, capital Y, it also produces white color. It means that this W gene, the dominant gene W, it masks the expression of the another dominant gene Y. So we can say that this is the case of the dominant epistasis, because the capital W, this is the dominant gene, and it has suppressed the expression of Y gene. In the presence of this dominant gene W, this Y gene, it is not able to express. That is why the color of the fruit is white. While as when this dominant gene capital W is absent, then Y gene expresses itself and the fruit color is yellow. So we can say that this the dominant gene, W dominant gene, it masks the expression of the another dominant gene Y, This is the case of dominant epistasis. And in the absence of this dominant gene W, this Y gene, it expresses itself and the fruit color produced is yellow. While as this double recessive, when there is double recessive condition, small w, small small y, then the fruit color is green. So we have seen that this dominant gene W, it masks the expression of another gene, that is Y gene. This, if we'll take W small, and if we'll take this condition, this is also white color, white fruit color. Here the dominant gene W, it has suppressed the expression of the recessive allele small y. So still the white color is produced. So ultimately what we will understand from this, that sometimes one dominant gene, it inhibits the expression of the other gene. It can inhibit the expression of the recessive allele as well as dominant allele. This is the case of the dominant epistasis, because dominant allele of the gene has suppressed the expression of the other gene. So this was in case of cucurbita pippo. Thanks.